In this video, we will continue our discussion about the CD8 plus T cells from the aged environment or in the young environment. So, in the panel H, they compare the differential expression genes of these young cells in the aged environment, and they are able to see that the B cells is significantly higher, whereas T cells and myeloid cells are low. Whereas when they checked for the reverse condition, that is differential expression genes in the aged cells in the young environment, they are able to see that in the T cells there is no significant difference, but B cells goes down and the myeloid cell goes up. So there is a difference in the B cells and the myeloid cells in the different environment, in the young environment and the aged environment, whereas not so much difference in the T cells. And when they go some deep into this differential expressions genes, they are able to see that almost similar number of genes are upregulated and downregulated in the young CD8 plus T cells versus aged CD8 plus T cells. And these things they have further checked when they do the analysis from the origin of the cells, that is CD45.1 in the young cells and CD45.2 is the aged cells, and they are able to see that the T cells differential expression genes is higher for the heterochronic conditions for both these aged cells and also for this young cells. So this is consistent. This is inconsistent with this panel J, that shows that whenever there is a heterochronic conditions, the differential expression genes goes up for the T cells. So afterwards, they wanted to see what are the transcription, what are the genes that are upregulated and downregulated in these young conditions and in the aged conditions, and they are able to find out that this Granzyme is usually, it seems to be downregulated in the heterochronic conditions compared to the isochronic conditions in the aged cells, whereas the naive CD4 plus related genes, that is LEF1 and SELL, goes up in the heterochronic conditions compared to the isochronic condition in the aged cells. And so this indicates there is a change, or there is a differentiation. There is a differentiation occurs in these aged cells when they move from aged conditions to the young conditions. So these things, moving forward, they also checked the pseudotime trajectory, and they are able to find out that the trajectory from CD8 plus naive cells to the effector memory T cells is observed for the aged CD8 plus T cells, indicating that they are more prone to move toward this effector memory T cells. And these things are further validated in this from this heat map, where we can see that the naive CD4 plus T cells is seems to be goes down when they move from this isochronic conditions to this isochronic conditions to these heterochronic conditions. That can be seen there. It seems to be goes down, and at the same time this CD8 plus T cells or effector memory CD8 plus T cells seems to be goes up when they move from isochronic heterochronous conditions to the heterochronous conditions, indicating that there is a differentiation occurs in this effector memory in this aged CD8 plus T cells from this naive CD8 plus T cells to the effector memory T cells. And from the violin plot, this is further these previous two results are further confirmed from this violin plot, where we can see that in the heterochronous conditions, this naive CD4 plus T cells population goes down in the aged cells, whereas in the case of for the effector memory CD8 plus T cells, this population goes up in the heterochronous conditions. So, and in this panel O also, for the heterochronous conditions, they are able to see that there is a decrease or lower amount of CD4 plus T cells are found, and the high amount of effector memory CD8 plus T cells are found. So this all indicates that in the case of aged CD8 plus T cells, they are more prone to move toward this effector memory T cells, which are usually less effective, or we can say that it is close to the this exhaustive CD8 plus T cells. However, that thing is, the last 2 points are not mentioned in the paper. These are based on my observations, or based on my understanding about this CD8 plus T cells. So that's all for this video, and if you need to understand more about this experimental results in detail, you can go through the reference that has been provided in the description. So thank you for your time.